Hello and welcome to another lecture from the Neuroimaging department at King's College London. This particular lecture is aimed at introducing the basics of positron emission tomography, or PET, as we will call it from now on. First of all, a reading reference. This is the book we have published, which is specifically designed for first-time users that need to learn all the basic notions that are required for building a PET experimental plan, executing it, collecting the data, and analyzing them. So this is the structure of the talk. First of all, we will try to understand what is the unique advantage of PET compared to other modalities in the context of functional imaging. Once we have established the need, we will start looking with some detail at the constituent parts of PET, that is, isotope production, radiochemistry, instrumentation, and the various computational aspects. And finally, we will look at how one can quantify biological parameters of interest using PET in three paradigmatic functional applications: cerebral blood flow, enzymatic rates, and receptor densities. So, why do we need PET? In order to answer these questions, we first need to, first of all, clarify why we need functional measurements at all, understand how one can measure a physiological parameter irrespective of the measurement modality, and how this can be achieved using tracers. 3, we need to define the principles for tracer kinetic experiments. 4, we need to check how these principles are adhered to by imaging modalities such as CT or MRI. And 5, then we need to explain why these modalities are excellent when measuring structural elements, but have limitations when employed in functional paradigms. And finally, 6, we need to provide a clear definition of PET and explain why it fully supports tracer functional measurements. So, why is it important to measure brain function in the context of mental health? The first reason is that brain disorders may not necessarily show structural deficits. As an example, addiction does not generally demonstrate any specific brain structural change. The second reason is timing. Functional changes generally precede structural changes. For example, in the case of Alzheimer's disease, ApoE4 carriers who have an elevated risk for the disease demonstrate changes in blood flow that precede by more than 20 years clinical overt symptoms. And finally, the third and possibly more important reason is that functional measurements allow the understanding of the pathological mechanisms involved in the disease and direct the development of treatments. So, once we have established that we need to make functional measurements, how do we go about doing them? We can do functional measurements using a tracer, the small red drop that you see in the picture. This drop will have certain biochemical properties that will make it move around body compartments that you see as these cylinders in the picture, which will move according to certain rates. Compartments can be either distinct physical tissues, say blood, the extravascular space, cells, and so on, or they can be distinct chemical states, say the original compound, its phosphorylated metabolites, and so on. The transfer rates across compartments are what we call physiological parameters. Typically, they can be things like perfusion or enzymatic rates, or from these rates we can derive, for example, other interesting parameters like target density or affinity. By following the red drop over time with the appropriate mathematical model representing the compartments, we can work out what these rates are. There are, however, 3 strict requirements for tracer measurements. The first one is that the tracer has to be a drop, meaning has to have a very small mass. in order not to affect the system we are measuring. 2. The drop will move across compartments in a well-comprehended and defined way. We do not use tracers to explore systems we do not know, but we use tracers only to calculate rates between compartments that we know well. And third, we need to have instrumentation able to measure the concentration of this drop through time. Now, a typical tracer is made by two components. One is the substrate that possesses the biochemical properties of interest, and the second one is the label that will make this trace visible to the measuring system. Now, can we use a CT scanner to image functional processes? A CT scanner works using X-rays that are emitted and then collected by the instrumentation itself. Hence, the scanner produces maps of X-ray absorption. It's a very accurate and quantitative technique, although resolution is not that high, nor its signal to noise is that great. However, it is also commonly found in any hospital. To use it with tracers, we need to have labels that are X-ray opaque. Molecules like iodine have this property. Let's look at MRI. MRI is a technique that comes to mind easily. It measures the relaxation of water proton magnetization. It produces images of excellent resolution and quality, and is not radioactive at all. I hope you recognize that profile because that nose is mine. Paramagnetic labels such as gadolinium are commercially available. However, there are problems with the use of these technologies with tracers. Molecules like iodine and gadolinium are quite big and change the biochemical properties of the substrates they are attached to. Importantly, they do not cross the blood-brain barrier. In any case, in the case of MRI, the amount of tracer will have to be substantial to be detected over and above the water proton signal, with likely interference with the targeted system. MRI is also not that quantitative, in the sense that it is not straightforward to translate the MRI images into images of tracer concentration. Hence, generally, both CT and MRI do not adhere to the first and third principles of tracer measurements. PET uses as labels positron emitters, such as oxygen-15, carbon-11, and fluorine-18. Positron is highly energetic, so only tiny amounts of label are really necessary to generate emission signal that can be captured by a scanner. Positron emitters are natural molecules that can be directly incorporated into the substrate of interest. This in case of, say, oxygen-15 and carbon-11, or these emitters are really small, like fluorine-18, and they do not change the substrate's properties. PET instrumentation is built to allow the accurate quantification of radioactivity concentration in the image, but also in the circulating blood. Hence, PET technology is absolutely designed to accurately fit the three tracer principles. So, let's now look at PET in some detail, starting from the production of the tracer. This is done in two parts. First of all, the radioisotope has to be produced, and then has to be attached to the substrate using radiosynthesis. We start with the positron. This, in the slide, is Paul Dirac, and the one on the board is his equation that was meant to introduce relativistic effects into the hydrogen model, but unexpectedly produced the prediction of an antimatter particle, the positron, a positively charged particle with the same mass of an electron. And This, on the top right side, is the picture of the first experimental verification of the positron by Carl Anderson at Caltech. The positron can be emitted by proton-rich nuclei. When released by decay, the positron travels a distance and then annihilates with an electron, generating two gamma rays, each of 511 keVs, traveling in exactly opposite directions. Now, PET centers generally have a cyclotron, a particle accelerator. The particle can be an ion or a charged particle such as a proton that is injected into the cyclotron in its central space. The two halves of the cyclotron have opposite charge, and the charge is alternated so that the particle is accelerated and starts rotating until it reaches the expected energy. The particle is then shot into what we call a target, which is a lead-shielded container filled with a gas. Different targets exist based on different needs, but one of the commonest gas used is nitrogen with a tiny percentage of oxygen. The nuclear reaction in this specific case sees nitrogen, atomic number 14, hit by a proton and generating an alpha particle, which is made of 2 protons and 2 neutrons, plus carbon-11, which is the carbon that is rich in protons, hence is an isotope emitting positrons. Carbon-11 attaches immediately to the oxygen in the mix, making carbon-11 CO2, which is a very reactive substance that can be readily used by the radiochemist to attach carbon-11 to a substrate. This part of the slide shows radioisotopes that are positron emitters. The percentage of the decay by positron emission is shown in the column designed beta plus decay. Together with their half-life and the substance they decay to. So for example, carbon-11 decays to boron. Note that for isotopes with short half-lives, such as oxygen-15 or carbon-11, the half-life is so short that the cyclotron has to be located really close to the PET scanners. Isotopes with longer half-lives can instead be shipped to the PET scanners from other locations. So, now we have the isotope and we need to do the radiochemistry. What substrates are we going to use? Well, the substrates for radiotracers are not that different from any drug. They need to be specific for the target and do not bind too much to anything else. So, for example, very lipophilic compounds tend to bind to lipids, such as those in cell membranes, which is not really good. However, they also need to cross the blood-brain barrier, and here, being lipophilic helps a little bit. They don't need to stick too much to plasma proteins, and they also need not to get metabolized too quickly by the liver. They also need to have the right kinetic, which means they need to get to the target and exit the organ, the tissue, during the experimental time, which is a maximum of 2 hours, really. A good example is the carbon-11 labeled RO154513. This is a drug that was developed by Roche in the 80s that acts as a competitive antagonist against the GABA receptors and can therefore be an antidote to the acute impairment caused by alcohol. So one could go to the pub, get drunk, pop up a pill, start the car and drive normally, only to crash an hour later as the drug was cleared very quickly out of the brain. So it wasn't really a good drug, but is an excellent PET tracer to measure GABA function, as in 1 hour or 1 hour and a half the tracer can go in and track the whole path and then exit. So now that we also have the radio tracer, we need the right instrumentation. Let's look at the ideal detection system. The label decays, generating the positron that annihilates with the electron, giving the gamma emission, the two gammas. So we need a ring of detectors made of materials that can stop the gammas and transform that energy into an electric pulse once the two gammas are detected. But how does the scanner know whether the gammas come from the same annihilation? Well, PET scanners use timing circuits. If two gammas are detected in the same narrow time window, we're talking of less than 10 or 5 nanoseconds, the two gammas will be paired. So that seems to be pretty straightforward. However, this is not reality. 3 are the problems of gamma detection that require corrections. 1. The first problem is attenuation, as gammas are absorbed by the tissue. Attenuation is corrected by building an attenuation map before tracer injection. This map can be obtained using rotating gamma sources. All PET scanners used to have them, or can be obtained from CT maps or MRI maps if one is using a PET CT or a PET MRI scanner. The second correction is scatter. Gamma rays interact with matter and change direction, creating a false echo. Scatter correction is made by using the attenuation maps to computationally simulate a scatter profile, generally. The third correction tries to estimate the so-called random events that occur when two unrelated gammas are detected in the same time window and erroneously coupled. This correction is also achieved generally by simulation, artificially scrambling events collected during the acquisition. Now that we have corrected the data, we suddenly realize that we do not really have an image yet. All we have is the detection of the gammas that are originated from the positron annihilations. These data are called projections. So how do we get from the projections that you can see in the slide to the original image? There are 2 methods to reconstruct images from projections. The first one is quite simple in principle. It consists in projecting back on the image the projected data. This recreates the original image plus stripe artifacts that are eliminated with some filtering. In fact, this technique is called filtered back projection. The second approach is a bit more complex. First of all, it consists in a computational model of the scanner that can recreate artificially, with great accuracy, the projections of a radioactive object in the field of view. Hence, the algorithm starts with some initial guess of the object, like the one you see here on the left, uses the computational model to generate the projections that are then compared with the real data. If different, the hypothetical image is then changed in some clever way, and the simulated projections are generated again. The process is repeated until the simulated object generates projections that are pretty close to the original data. This method is called iterative reconstruction. Filtered by projection is quick and very quantitative, but as you can see over there, the stripe artifacts are not entirely eliminated, and they disturb the visual assessment of an image, particularly when the distribution of radioactivity is quite heterogeneous. Iterative reconstruction images instead have great visual appeal, but the technique is very slow to converge, particularly for small radioactive objects. Noise instead is very different from filtered by projection, as it is quite speckled. Let's now look at data collection and how the data are used to quantify physiological rates. What we see in the slide is the full PET experiment. More simplified designs are also adopted for more mature or clinical applications. The study generally consists in the injection of the radiotracer IV followed by a dynamic PET measurement that also consists in a series of frames acquired over a period of time. Could be 60 minutes, 90 minutes or more. At the same time, in order to calculate the concentration of the radiotracer in plasma, in the capillaries obviously, which is the input that we need, the radial artery is cannulated and blood is drawn for the duration of the study. Note that the concentration of the compound at the peripheral artery will be the same as the one in the brain capillaries. Also note that the radioactivity in blood will have to be corrected for radioactivity in things like blood cells, red cells, plasma proteins, as well as for the radioactivity generated by the radioactive metabolites of the radiotracer that have been processed peripherally, generally by the liver. Once the plasma and the imaging data are ready, they are used into a mathematical model that mimics the compartments that we have seen before, and through this model we can calculate the rates we were looking for. These calculations can be done for brain regions or can also be done at the level of the pixel. In this case, the output of the whole procedure will be what we call a parametric image, an image where each voxel contains the value of the physiological parameter for that location, as you can see in the bottom left of the slide. In this slide, we see how the qualitative description of the tracer, top left, is then translated into a mathematical model, which is bottom left, that uses the plasma input function and the mathematical description of the compartments. The rates of this model are then adjusted by an optimization algorithm so that the simulated activity in the tissues fits the real data. But what are these compartments? As discussed before, a compartment is an idealized container of a substance that can either represent a segregated state of the tracer or a biochemical transformation. The equations that are used are actually quite simple, and they are written as mass balance equations. Say the change in the quantity of a substance in a compartment is simply equal to what gets in minus what gets out. So, let's now try to make some nice examples. And the first, a paradigmatic one, is cerebral blood flow. It has been known since the end of the 19th century and the work of Sherrington that the brain has its own local control of blood flow. Before that, it was thought that the heart was controlling the brain. In other words, it was the controlling station for the whole body. However, for a long time, scientists struggled to find a way of measuring cerebral blood flow. Seymour Katy and Carl Schmidt were the first to work out a methodology that was then called the Katy-Schmidt method for the measurement of cerebral blood flow. These two are giants in the history of medicine. Seymour Katy, a junior at the time, went on to prove the genetic basis of schizophrenia and became the head of the National Institute of Mental Health in Bethesda. Carl Schmidt was already well known at the time as, amongst other things, was the first physician in the US to encounter the Great Flu pandemic in 1918. The Katy-Schmidt method is described here. The subject was resting on a table breathing nitrous dioxide. Nitrous dioxide is an inert gas that is entirely extracted by the brain. It does not do anything and is then cleared to the venous system. From the picture you can see that there were two needles inserted. One in the radial artery to sample the gas concentration in arterial blood, the second one in the jugular vein, which makes this clearly an invasive methodology. The dynamic of the gas is described by the equation that says that any change in the gas concentration in brain depends by the rate of entry, K1, and by the rate of exit, K2. K1, which is also called the tissue perfusion, is equal to the extraction E times the cerebral blood flow. Now, For nitrous dioxide, E is actually 1 because it is fully extracted. Now K2 is equal to K1 divided by the volume of distribution of the gas in the tissue, which is actually known and fixed. At the time, there was no imaging method, so they had to use the venous concentration as a proxy for the tissue concentration, because the two get in equilibrium, say, after 10 minutes. They could then solve the equation, fitting the data to obtain K1. And because E was 1 and known, they could obtain the cerebral blood flow estimate. Note that this was only a global brain measurement. In any case, this method was widely used. For example, it led to the discovery of REM sleep. Scientists at the time noticed that while cerebral blood flow decreased over sleep, a few individuals showed higher than usual values. And this happened because they had been measured during REM. The advent of PET simplified everything. This time, they needed not to use a gas, but a fluid, water, which is inert and is 100% extracted by the brain. Water was labeled with oxygen 15 and injected, and its dynamic concentration through time in brain and plasma was measured. The data were then used into the Katy-Schmidt equation to produce, bottom left, pixel by pixel images of cerebral blood flow. So now let's move to another paradigmatic example. An example for enzymatic fluxes, and in particular glucose metabolism. The oxidation of glucose is by far the main source of energy for the brain. The reaction is shown in the slide and looks really complicated. Let's start from the top, where glucose is sitting in plasma and is then transported across the blood-brain barrier into the cell. This is a process facilitated by glucose transporters. When entering the mitochondria, glucose is then phosphorylated to glucose 6P, which makes the molecule much more reactive. At this stage, three things can happen. Glucose can be stored as glycogen, but this doesn't really happen in brain, or is diverted into the pentose phosphate shunt, where it is used to produce nicotinamide adenine dinucleotide phosphate, which is used to fight oxidative stress. However, the large majority of glucose is used in glycolysis to produce pyruvate that is then used in mitochondrial respiration to produce ATP. Oh wow! Can we label glucose and follow all these complicated steps? The answer is, well, no. However, it turns out this is actually not necessary. The cell controls the metabolic rate of glucose by adjusting just the first step, the first rate, the phosphorylation rate. So phosphorylation, glucose phosphorylation, is the only process we really need to trace. As it happens, there is a glucose analog, which is called deoxyglucose, a glucose without the OH group, that does exactly that. It is transported into the cell and then is phosphorylated like glucose, but then it stops there and it accumulates in the mitochondria because nothing else happens to him. In fact, because the accumulation will stop any other glucose to entry, deoxyglucose is a poison. But if used in small quantity, it just works fine. This is the idea behind the Deoxyglucose method to estimate the metabolic rate of glucose proposed by Louis Sokolov in the 70s. Deoxyglucose is injected and measured through time, both in plasma and in brain. As there was no appropriate imaging in the 70s, this method only applied to animal experiments, where rats, mice were sacrificed at different times. The data were then used in the model to calculate the three rate constants: K1, transport, K2, efflux, and K3, phosphorylation. Then the total flux of glucose can be calculated as the product of the rate Ki, which, as you can see from the slide, is directly derived from the rate constants, times how much glucose, not deoxyglucose, glucose, you have in plasma. which is a straightforward measurement. One more thing, lambda is called the lumped constant. Is a constant, doesn't change, which accounts for the difference in affinity of the glucose transporter for deoxyglucose compared to glucose. Once PET was available, it was straightforward to move the technique into PET, imaging of glucose metabolism. Deoxyglucose was labeled with fluorine 18, and once injected, the radioactivity in blood and brain was monitored for some time, say 60 minutes, maybe 90 minutes sometimes. The data were then used into Sokolov's model to produce pixel by pixel maps of glucose metabolic rates. Now, you should have the hang of this. Let's now say we can quantify receptor densities, the third paradigmatic application in mental health. The idea, pioneered by Mark Minton, is quite simple. We choose a drug that targets the receptors that we are interested in. We label it, generally with carbon 11. Then we inject it, and the radiotracer will go into brain tissue, which is the free ligand you see in the slide. And then we'll attach and then detach to the receptors with a certain rate. From these rates, the K3 and K4 rates, we can directly infer the density of those receptors. So that's the end of the lecture. Thank you very much for tuning in.